I'm Camila, and today we will be going over DNA replication. So DNA is structured like a ladder, with deoxyribose sugar and phosphates connecting with each other to make the sugar-phosphate backbone. The end of a strand of DNA with sugar is the 3' end, and the end of the strand with phosphate is the 5' end. There are 4 nitrogenous bases which connect both strands. Thymine and adenine have 2 hydrogen bonds between them, and guanine and cytosine have 3. Both adenine and guanine are purines, which means they have 2 rings, and thymine and cytosine are pyrimidines, which means they have 1. Lastly, these DNA strands will always be in an anti-parallel structure. So DNA is normally wound up in our cells, so we have to go through a certain process so we can undo it to be able to replicate it. The enzymes helicase and topoisomerase do this job. I might have not pronounced that right, but oh well. Topoisomerase goes across the strand first, and as you can see, it starts loosening up the coil and the DNA strand as a whole. Helicase is the enzyme that ends up separating both the strands completely, breaking their bonds and undoing the strands. This happens in multiple places across the strand at the same time at origins of replication. And these processes only occur in the 5' to 3' direction. These DNA strands will make the DNA unwind faster so we can go through replication faster. As the DNA strands separate, single-strand binding proteins hold them apart. This makes it so 2 DNA strands can be made out of 1. Each separate strand is a template for the new DNA. One of the separated strands is the leading strand and the other is the lagging strand. The leading strand is the strand that has the 5' end first, and the lagging strand has the 3' end first. Now we will focus on the leading strand. Since replication happens in the 5' to 3' direction, the leading strand has continuous replication. An RNA primer attaches to the strand first, which initiates DNA synthesis. DNA polymerase III actually creates a new strand and attaches cytosine to guanine and adenine to thymine. DNA polymerase I then removes the RNA primer, so you have a new DNA strand that's being made. Now you get to watch me do more arts and crafts. Yay! As I'm building the second model, I gotta tell you, I know, I'm tired of the music too. It's just what came in the program. Now for the lagging strand. The lagging strand replicates discontinuously because, as I said before, DNA replication can only happen in the 5' to 3' direction, and this one starts with a 3' end. Like before, an RNA primer attaches to where DNA polymerase III should start to replicate the new strand. In this case, multiple primers have to be used since there is discontinuous replication. Each separated replicated part is called an Okazaki fragment. The lagging strand uses ligase to connect each of the Okazaki fragments. Then DNA polymerase I does the same thing and removes the RNA primers. The energy to make a new DNA strand does have to come from somewhere, though. It comes from cutting high-energy phosphate bonds in the DNA. So that energy is released to create a new strand. Now for the big completed model. Eventually, all the little steps that I explained to you will lead to 2 new completed DNA strands. And that concludes DNA replication by me.